Hello, everyone, and thank you for being here. I'm honored that Professor Laszlo Boros has invited me to participate in this exciting conference. And I hope you will find what I have to say today to be stimulating and exciting and probably a bit provocative. So let me begin with my title, COVID-19, Glyphosate, Deuterium, and SARS-CoV-2 vaccines. A lot of topics are being covered here. I want to start with a quote. We have never seen this before. However, the vision is so horrible and so awful and so terrifying that I don't even want to know what happens next. And this is Professor Sucharit Bhakdi, who's a retired microbiologist, and he's speaking about the vaccines against COVID-19. So here's an outline of my talk. I'm going to do about 1/3 of the talk on the disease itself and its connection to glyphosate toxicity. Glyphosate is the active ingredient in the pervasive herbicide Roundup. And then the second part will be 2/3 of the talk, talking about the COVID-19 vaccines, concentrating on the messenger RNA vaccines, because those are the ones I know best, although I think the DNA vector vaccines are also toxic. So in part one, I'm going to focus on the immune cells, their mitochondrial defects that are caused by glyphosate, and how that impacts COVID-19 disease consequences. I'm going to emphasize the importance of NAD/PH redox system to mitochondrial health, and talk about how excess deuterium in mitochondria disrupts their function. I'm going to argue that this infection with SARS-CoV-2 causes a very interesting response in the lungs through the immune response that aims to restore mitochondrial health by restoring their low deuterium and by providing the immune cells with extra mitochondria. And this will be then the topic of rebooting mitochondria, the third subtopic of this part. In part two, I'll talk about the COVID-19 vaccines, an overview, particularly of the mRNA vaccines, the technology, and why I suspect that it has negative consequences. I'm going to emphasize exosomes and microRNAs. Exosomes are small lipid particles that are produced by cells throughout the body, released into the circulation, and they serve as a communication network between one cell and another. Very, very fascinating space that I've gotten very interested in recent times, as well as the world has gotten interested in exosomes due to their ability to deliver things among cells. They carry in particular microRNAs, and the microRNAs are really important signaling molecules. They're short sequences of nucleotides that can bind to the promoter of the gene for various proteins. So each microRNA has specific functions. And there are 3 particular microRNAs that I will focus on in the discussion that have been shown to be connected to COVID-19. And then they have very, very interesting consequences relating to heart disease and prion diseases and other things. I will have other consequences of these microRNAs in a third section. So there's 3 sections here on those microRNAs. And then I'm going to specifically talk about the spike antibodies and their potential to cause autoimmune disease. And finally, is reverse transcription and the potential for the spike RNA to become DNA and to become a permanent part of human DNA. So part one, defective immune cell mitochondria and COVID-19. First, the big picture. Deuterium is a natural isotope of hydrogen, has an extra neutron, a proton, an electron, and a neutron, makes it twice as heavy, makes it have different biophysical and biochemical properties. Mitochondria depend on low deuterium in their water to function well. And the supply of deuterium-depleted water to the mitochondria depends upon Nicotinamide Adenine Dinucleotide, NAD, as a proton carrier. It's a central molecule in metabolism and heavily used by the mitochondria to supply deuterium-depleted protons to their water. Glyphosate disrupts the supply of DDW to the mitochondria via NAD. Defective mitochondria in the immune cells causes immune deficiency, which means that it's hard to clear the virus when it infects the lungs. And that leads to a profusion of viral growth, followed by an extreme reaction of the immune system, and then causes the cascade that leads to disease. It launches a cascade response and the purpose of that response is to restore mitochondrial health to the immune cells. I will argue this, and we'll see if you can agree with that. Once the macrophages are reinvigorated, then they can clear the virus and the disease heals. If the immune system is simply too sick to fix, the person dies. So the importance of NAD/PH redox system to mitochondrial health. The big picture, NAD is an essential cofactor in the oxidation reduction reactions, and there are many of them throughout the body. It exists in 4 forms, with and without hydrogens and with and without phosphate. So NAD+, no hydrogen, no phosphate, NADH, NADP+, NADPH. And the enzymes that supply NAD+ and NADP+ with the H are all specially designed to avoid deuterium. They have very special properties. Many of them are called flavoproteins. They're able to select hydrogen over deuterium, and they're able to even make sure that what ends up in the product is hydrogen, even if what started on the source was not. The mitochondrial organelles inside the cell that supply energy in the form of ATP. They depend critically on a proton ATPase pump to make that ATP, and the protons derived from NADH and NADPH are pumped into the intermembrane space. So deuterium gums up the pump like sugar in the gas tank, makes it work badly, makes it produce less ATP, makes it release reactive oxygen species that damage the mitochondria. And glyphosate, which is Roundup, interferes with the supply of NADH and NADPH to the organism. So here's are the arguments that I have for how glyphosate impairs these pathways. These arguments are all developed in my new book, Toxic Legacy, which is available for Chelsea Green Publishers, just released in July of this year. And so you can refer to that book to get the details on this. But glyphosate is the active ingredient in Roundup, kills weeds by suppressing EPSP synthase in the shikimate biological pathway. The argument is that our cells don't have this pathway and they don't have this enzyme, and therefore we're safe, but that argument is flawed. Our gut microbes, many of them have the pathway, have the enzyme, and get disrupted by glyphosate. And a major product of that enzyme is tryptophan. And tryptophan is a precursor to NAD. So when you have a deficient ability to make tryptophan by the gut microbes, you end up with NAD deficiency. Worse than this, glyphosate disrupts the enzymes that deliver the H to the mitochondria. It induces reactive oxygen species in mitochondria. That's been shown in many studies. It depletes glutathione. That's also been shown. It also increases the ratio of oxidized to reduced glutathione, indicating oxidative stress. And then specifically enzymes inhibit certain enzymes, and two of them that are important here are glucose-6-phosphate dehydrogenase and succinate dehydrogenase. And if you know chemistry, you know these two are very, very important. This one reestablishes NADPH. This H comes back through this enzyme and supplies it to the glutathione to keep glutathione in its reduced state. So when this is broken, this is broken. It also inhibits succinate dehydrogenase, and that's the only enzyme in the mitochondria that plays a role in both the citric acid pathway and oxidative phosphorylation. So this enzyme, these are both dehydrogenases, and in general, I think glyphosate disrupts dehydrogenases. They typically are flavoenzymes, and the mechanism is complex, but it's described in this book. COVID-19, so here's a paper that I was delighted to find this paper because it was so perfect, given what I was thinking. NAD plus deficiency may predispose the aged, the obese, and type 2 diabetics to mortality through its effect on SIRT1 activity. This is a hypothesis in this paper published in 2020. And so they talk about the fact that this is such an important cofactor for this proton-coupled electron transfer. These sirtuins are a class of ancient proteins that are NAD plus dependent. They won't work without NAD plus. And they're signaling proteins that regulate metabolism. So when intracellular NAD levels are depleted in association with these diseases, diabetes and obesity, which are also risk factors for bad outcome in COVID-19. And these two diseases are two of the many diseases that have been rising in prevalence in the United States, exactly in step with the rise in glyphosate usage on core crops. And this paper by Nancy Swanson et al. Has over 30 figures in there with various correlations between all kinds of diseases and the rise in glyphosate. Depletion of SIRT1 because of this NAD deficiency causes uncontrolled increases in the inflammatory markers. These things are shown to be very highly expressed in the cases where COVID-19 gets out of control. In the lungs, these are all inflammation-inducing agents that cause this extreme immune response that ends up destroying the lung tissues. And that's called a cytokine storm. And they're arguing that it's due to this inability to activate SIRT1. So very interesting paper, very interesting concept. I happen to think that it makes a lot of sense. Now we get to the infection in the lungs and how does it unfold and what is its purpose? I always think that everything that happens in biology has a purpose beyond just making you sick. So when you get sick, you have symptoms, but something good is happening in terms of trying to restore a basic level of health to the system itself. And so when you look at COVID-19, it's very interesting to look at the initial COVID-19 events in a case where it gets into extreme disease. The viruses come in through the bronchioles. They infect the epithelial cells in the lungs, in these alveolar airspace. They infect the epithelial cells here. And then the macrophages come in, but they fail to clear the virus because they're defective. This is the argument that I will make. And I have a whole chapter in my book on glyphosate suppression of the immune system. The viruses proliferate wildly. The macrophages send alarm signals, which draws in more macrophages and many other immune cells, including platelets and stem cells from the bone marrow. They all come in to assist in the problem. The capillary wall becomes leaky to support the invasion. Blood pressure drops, fluid begins to fill the alveolar space, and the person feels like they're drowning. So now here's a paper, is a bradykinin storm brewing in COVID-19. Very interesting to link bradykinin to this crisis because it does look like that's a major part of what's happening. Hypertension is a risk factor, but hypotension develops instead during the disease process. And now these ACE2 receptors are important because the virus binds to them. And that's part of the mechanism by which it causes disease. They're upregulated by 199-fold in the lungs in severe cases. And then ACE is downregulated. And the effect of that is that ACE would normally clear bradykinin, but because it's suppressed, the bradykinin builds up. The receptors are upregulated by a huge amount, 3,000-fold, all in this disease process. Sorry about that. So bradykinin, they're upregulated. Bradykinin induces vasodilation and hypotension. And these inflammatory cytokines induce the capillary leakage, inhibit alveolar fluid reabsorption, and that gives you the flooding with the water. So now here's another one, bradykinin-induced hyperpermeability. This is the same paper, I think, of the lung capillaries, causes the formation of hyaluronic acid hydrogel. This is very interesting to me, and I'll say why in a moment. It inhibits gas exchange, so it's going to make them feel like they can't get oxygen, which is in fact the problem. Here's your gel. This is like a jello caused by the hyaluronic acid. And then you have vascular hyperpermeability, downregulation. So these two work together to make this hyaluronic acid build up. Inflamed membrane inflammation, all throughout here, inflammation. And this is where the action is, I think, where the deuterium depleted water is being created to restore mitochondrial health. We'll get to that in a moment. SARS-CoV-2 causes massive overproduction of hyaluronic acid in the lungs. It can trap roughly 1,000 times its weight in water. And when it's bound to water, the resulting hydrogel obtains a stiff, viscous quality similar to jello. This is important. Multiple enzymes that synthesize it are massively upregulated, 9,000-fold for HAS1, and these two are also upregulated. Excess hyaluronic acid is associated with pulmonary thrombosis, these ground glass opacities that they observe, and acute respiratory distress syndrome. And then this could form a viscous hydrogel that would negatively impact gas exchange, and that's true. However, I think it's forming another role as well. And this is my hypothesis that the virus is actually facilitating the repair of the deuterium problem in the mitochondria. The hydrogel traps deuterium, which leaves deuterium depleted water in the fluid water that's left behind. The virus contains lipids such as linoleic acid in its membrane. They're stolen from the host cell as the virus escapes the host. It has these little pockets that fit perfectly with the linoleic acid. The inflammatory response to the virus, which is a consequence of the weak immune system, causes the release of lipoxygenase. And lipoxygenase is an extremely interesting enzyme. What it does is it extracts two protons from a lipid, such as this linoleic acid, and it combines them with oxygen to make deuterium depleted water. And lipoxygenase is by far the strongest enzyme I have found in the literature in terms of its what's called a deuterium kinetic isotope effect. It has a fantastic ability to favor hydrogen over deuterium. And also these lipids are already reduced in deuterium to begin with. And this thing just basically makes sure that this is no deuterium in this water that's being produced through the lipoxygenase action. It produces these molecules called leukotrienes, which are actually signaling molecules that induce further inflammation. So it's kind of a positive cascade, but the whole point is to get that high quality water to be able to fix the mitochondria in the macrophages. The arterioles constrict, the venues open up leaks, the water comes in that's being made in the capillaries, and the macrophages essentially drink the sweet nectar that's supplied to them, and they replenish the mitochondrial water in their mitochondria to help fix their problem. And now, interestingly enough, viruses are stabilized with excess deuterium. I was really surprised to see this. The protein in SARS-CoV-2, the nucleocapsid protein, starts to unfold at 35 degrees centigrade, which is just below the normal temperature of the body. If you get up to a higher temperature, it completely degrades. So a fever actually helps to break down the virus. And that may be one of the most important reasons for a fever, to try to clear the disease. They take up deuterium, they trap it in their protein coat, and they also trap it in their single strand of RNA. So again, they're removing deuterium from the fluid water. And the deuterium stabilizes the viral protein and the RNA and protects from temperature denaturation. So the virus removes deuterium from the body fluids. But when the deuterium level of the body fluids is high, the virus is going to be more stable. So that's going to be another factor that's going to increase the risk of viral infection. Okay, rebooting the mitochondria, very interesting. I started to look into the whole idea of cells sharing their mitochondria, and I was quite surprised by what I have found. I've now read many papers from the research literature on this topic. Various immune cells and platelets come into that space where all this inflammation is happening, and they let go of their mitochondria. They release them in various forms, in various ways, even just straight mitochondria into the water, mitochondria inside exosomes, mitochondria in these little tubes that we'll talk about in a moment that can transfer them directly from one cell to another. And so this one is talking about tunneling nanotubes. That's what is the topic here, tunneling nanotubes, which are so cool that connect up two cells. And it would be, for example, a mesenchymal stem cell that's come in from the bone marrow, which can transfer its mitochondria to the macrophages. And then the result is the macrophages are getting enriched in new mitochondria that they desperately need. So these stem cells are actually improving the bioenergetics. And this can be a novel mechanism of the antimicrobial effect of these mesenchymal stem cells. And so here's a nice picture of two cells with a tunneling nanotube. These things are so cool, and lots of things can go through these tunnels, which includes, so you can have the tunneling nanotubes can transport whole organelles, mitochondria, endosomes, lysosomes. These things are also often in trouble in these defective states. So they're getting new versions of mitochondria, endosomes, lysosomes, and there can be other things like viruses and prions, which we'll talk about in later on with the vaccine, and calcium signals. So all these things can be transferred from one cell to another. And typically from something like a mesenchymal stem cell to a macrophage, it needs to get boosted so it can cure the disease. So the exosomal transfer of mitochondria from airway myeloid-derived regulatory cells to T cells is a paper here, which is quite interesting, where these are like from the bone marrow. These cells come in, infiltrate the inflammatory site. And this is, for example, in asthma, this was before COVID-19, this paper. In these MDRCs, transfer mitochondria to T cells via exosomes. And these exosomes are these small lipid particles that are released by cells when they're under conditions of stress. They're coming out of the endosome in a situation where it's impaired and it can't get converted into a lysosome. So it makes all these little particles and releases them into the circulation and puts all kinds of interesting things inside them. We'll have a lot more to say about exosomes later. And then, but they can be exosomes inside, mitochondria inside exosomes that can be transferred to a cell and work in the other cell. They've been shown to still be functional when they go into the recipient cell. And so what I'm hypothesizing is that these immune cells can sweep up deuterium-depleted water and mitochondria via macropinocytosis, a beautiful word that is basically a big cell that just sweeps in the deuterium-depleted water and everything that's in it to help to replenish its supplies for its various organelles that are deficient. So platelets, I think, are very, very interesting because it turns out that they each contain 7 or 8 mitochondria. They don't have a nucleus. They're really stripped down, but they do have mitochondria. And there are lots and lots and lots of platelets. So they can come in and they release their mitochondria. And those mitochondria can be a very good resource to help the immune cells to replenish their own mitochondria. Activated platelets, they form blood clots. And when they form the blood clots, they release their mitochondria. Of course, the blood clots can lead to disseminated intravascular coagulation, which is something we are seeing in both COVID-19 and the vaccines, and ultimately multiple organ failure. So the mitochondria get released into the extracellular space from the platelets, and then they can either be freely released or they can be embedded in exosomes. And in any case, they can get taken up by the immune cells to replenish their mitochondria and restore their strength. Now, megakaryocytes are another interesting class of cell. They're huge cells that have lots of nuclei, and they make the platelets. And they're mostly in the bone marrow, but there's a whole bunch of them in the lungs as well. And the megakaryocytes in the lungs can go directly and release exosomes, bypassing the stage of platelets. So they normally make lots of little platelets and ship them out to the world. They could just directly make exosomes. And in those exosomes, they can release mitochondria that can help out the system. So they both release these extracellular vesicles, such as exosomes. There's various sizes of vesicles, but the exosomes are the smallest ones. And then this CD41 is a marker for both the megakaryocytes and the platelets. And this study was looking at the levels of expression of CD41 and as well as the extracellular vesicle release. And it's very interesting to look at the control. And this was a non-severe COVID, and this is a severe COVID. the statistics over the group of people who had non-severe COVID, you can see had the highest numbers of exosomes and from both megakaryocytes and platelets, much higher numbers even than the severe group. So it suggests that they had sufficient resource to do this. These didn't need it. This one failed because they couldn't produce enough. That would be my interpretation. And it's interesting that platelet counts over time can be monitored and they can be an indicator of the health of the patient. And this study looked at that for survivors versus non-survivors of COVID-19, looking over time, day 1, day 3, day 7, day 14. The people who survived tended to have significant recovery of the platelets. The ones who died started to lose the level of platelets down to the point where they died when the platelets were lost completely, or essentially down to very low levels. So summary of part 1, COVID-19 affects different countries to different degrees, and the primary discriminator could be glyphosate exposure. I think I have been seeing that many of the countries that are hit hardest by COVID-19 are the countries that also heavily use glyphosate, and countries that are mostly small organic farms have almost no problem with COVID-19. It's quite remarkable. Glyphosate disrupts the body's ability to properly manage deuterium, and then deuterium toxicity results in impaired innate immune function. The process that unfolds during acute COVID-19 aims to restore mitochondrial and lysosomal health to the immune cells. Inflammation, swelling, alveolar hydrogel, they all reflect mechanisms that produce deuterium-depleted water. Platelets and mesenchymal stem cells come in and supply fresh mitochondria to the macrophages via these tunneling nanotubes and exosomes. The macrophages use macropinocytosis to acquire both deuterium-depleted water and mitochondria. Eventually, it reboots the mitochondria to support viral clearance. Okay, part 2, the COVID-19 vaccines. The big picture. The messenger RNA COVID-19 vaccines have been carefully bioengineered to optimize for inducing high levels of antibodies to the spike protein. That is their singular goal. However, if you make too many antibodies, you can actually end up attacking your tissues through molecular mimicry and get autoimmune disease. The injection bypasses the mucosal barriers and the vascular barriers and goes straight to the muscle cells in the arm. And this raises alarm bells in the immune system. The spike protein is toxic and it's prion-like. And many studies have shown that it is the most toxic part of the virus. It's produced in large amounts by these in the germinal centers of the spleen as a response to the vaccine. And it gets distributed throughout the body via these exosomes. Those exosomes are toxic because they contain prion-like spike protein, as well as we will see microRNAs are packaged inside those exosomes along with the spike protein. Those microRNAs are signaling to the body that we've got trouble here because of this massive production of this toxic protein. The exosomes deliver spike and microRNAs to the brain to induce protein misfolding, which leads to all kinds of neurodegenerative diseases and also brain cancer. The prize of the vaccine is a retuning of the immune system policy towards autoimmune disease and neurodegenerative disease, sort of admitting a failure to be able to use the virus in the way that would normally be intended, making these antibodies that prevent the virus actually from infecting, but as a trade-off, you have chronic inflammation throughout the body. And that is, I think, another way to create deuterium-depleted water in these injured organs to help repair the mitochondria. That's hypothesis. Overview. This is a paper that I wrote together with Dr. Greg Nye, published in May of this year. Worse than the disease, reviewing some possible unintended consequences of the messenger RNA vaccines against COVID-19. This is a long paper, 42 pages. And our main point is that the vaccines are poorly evaluated, novel technology with many unknowns. And these are some of the adverse consequences that we identified as being plausible, and we gave the arguments for why we believe that, along with a large number of references. And most of this I won't have time to talk about here. I recommend reading this paper if you'd like to know more. Pathogenic priming, this multi-system inflammatory disease, autoimmunity, allergic reactions. This is through the polyethylene glycol that's in the vaccine. Many people have allergies to polyethylene glycol, and that gives them anaphylactic shock. Antibody-dependent enhancement is an interesting possibility. We've seen it in earlier vaccines against coronaviruses. Not clear yet whether this is going to happen with this one. We'll have to wait and see. Where you actually produce the antibodies, but they make the virus more dangerous rather than less dangerous. Activation of latent viral infections. We're definitely seeing that because we're seeing cases of Bell's palsy and shingles, and these are consequences of viruses that are usually latent that are getting waking up. And I think that's because the generic innate immune system is being weakened by the vaccine. neurodegeneration and prion diseases. I will have a lot to say about this topic in a moment. Emergence of novel variants is also interesting. They've been blaming the anti-vaxxers for all these new variants that are coming out. The vaccinated people are catching the disease from the variants. The variants are resistant to the antibodies. We predicted in our paper, and we wrote a section about the possibility that people who are immune compromised when they get the vaccine, they give the virus a training ground for recognizing what the antibodies look like, and then the virus can mutate its way out of a jam. The potential for integration of spike protein gene into human DNA is very interesting, very serious, potentially very unknown consequences of such a thing. Now, this paper is called the RNA life cycle, just as good for, I'm referencing it just for its illustration of how proteins are made in human cells. But here I have points about the vaccine itself, which you can learn about in more detail by reading the paper that Greg and I wrote. The vaccines are cleverly designed to basically fool the body into thinking that what this protein, what this RNA is a messenger RNA made by a human cell, ready to go to turn out lots and lots of protein. And the code that it codes for is the spike protein in the virus. And so normally you have the DNA, which gets mapped to RNA, and then the translation takes place and the protein comes out. You have to add some things like this 5' cap and this poly A tail. All of these are added to the DNA. The gene is coded in DNA initially, and then there's enzymes that convert it to the RNA, and then they modify the RNA by putting on these extra pieces that make it look exactly like a long poly A tail, which helps to protect it from breaking down. And then they also do lots and lots of manipulations. So this RNA is definitely not natural. They modify the nucleotides. All the uridines are replaced by methyl pseudouridines, all the uridines in the protein, because it was observed that that would help to keep the RNA from getting broken down. They're obsessing on keeping it from getting broken down and making it ready to go when it enters the cell to make lots and lots of protein. By replacing all the A's and U's, actually in the case of RNA, to G's and C's, that they can without changing the code, ends up producing a GC-rich nucleotide sequence, which has been shown experimentally to greatly increase its ability to turn out proteins quickly. So everything is designed to make lots of spike protein, last for a long time, be unable to be broken down. The PEG is going to stabilize the protein to keep it from being broken down. And then they add in some cholesterol and some phospholipids to make it look like a regular LDL particle, a lipid particle that's a natural thing that human cells produce. And that therefore it can take advantage of the natural mechanism by which these LDL particles are taken up. Now they put in these interesting cationic lipids that are also not natural. They're synthetic cationic lipids, especially chosen because they introduce, and this is a study that showed that they induced massive neutrophil infiltration, activation of diverse inflammation pathways, production of various inflammatory cytokines and chemokines. All of this is good because they want to make the immune cells come into the muscles, take up the messenger RNA, carry it into the lymph system, deliver it to the spleen, because the spleen is where you're going to make lots and lots of antibodies. And this is the goal of the vaccine, and it succeeds phenomenally at its goal. I would say that these vaccines are amazing in their ability to produce antibodies to the spike protein, which indeed does decrease the severity of the disease. So in a sense, they achieve their goal. But the question is, at what cost? So this is just showing cationic liposomes, positively charged lipids, cause all these problems with the cell. And they particularly work well as an adjuvant, such as the aluminum that's normally put in many other vaccines. So this was a really interesting 3-hour interview that I watched. I don't often watch these videos because they take time, but this one was really fascinating, 3 hours long. Dr. Brett Weinstein interviewed Dr. Robert Malone and Mr. Steve Kirsch, who's an MIT grad and an entrepreneur. And these two people really came on strong with their worries about these vaccines. And they complained about no evaluation of reproductive toxicity or genotoxicity in animals before the vaccines were authorized for humans. A FOIA request from doctors in Canada yielded a Pfizer study. It was written in Japanese, but you could look at the tables and you could see where the lipid nanoparticles went. And they found them in many parts of the body. But what was especially disturbing is where the highest levels accumulated. And Greg and I had identified the spleen from a previous paper as a place where they accumulate, but also the lymph nodes and the lymph nodes. But they also found the ovaries, the adrenal glands, and the bone marrow, in addition to the lymph nodes, the spleen, and the liver, which we talked about in our paper. So I was really surprised by this one. The ovaries, very concerning. Many women are complaining of heavy periods and irregular menstrual cycle in response to the vaccine. And I was just suspecting that if you're producing lots of spike protein in the ovaries, it's going to cause problems. There's also many, many miscarriages in the vaccine event reporting systems. Very big concern about pregnant women getting it. I would be very concerned about that, or about it affecting reproduction. Dr. Malone is a very impressive person. He was one of the early players in the messenger RNA vaccine technology, so he should know what he's talking about. And he's very concerned about these vaccines. So the germinal centers in the spleen is really where the action takes place. The RNA, the immune cells come to the muscles, which are screaming for help. Immune cells come in, they start taking up the vaccine particles. They take them into the lymph system. The lymph nodes get swollen under the arm. They take them down into the spleen. And then in the spleen, they go into the germinal centers, and that's where they really get down to business, turning out antibodies, which is the goal. They produce, so these germinal centers are specialized microstructures, and they, especially in the spleen and the lymph nodes, they produce long-lived antibodies secreting plasma cells and memory B cells, protecting against reinfection. And so these B cells undergo their somatic mutation process, which achieves selection of a particular antibodies that match to the spike protein, various pieces of the spike protein. And the B cell clones bind to antigen with high affinity. They emerge from this process, and you have succeeded. But however, this mutation process can also be dangerous because it can create autoreactive clones that can cause autoimmunity. So the SARS-CoV-2 mRNA vaccines induce persistent human germinal center responses. This is a paper about these vaccines that showed, in fact, that they did exactly what they wanted them to do, which was to go into the spleen, remarkable capacity to induce robust and prolonged reactions leading to memory B cells and clones and all of this stuff, the epitopes, everything perfect for producing antibodies to the spike protein. So exosomes, microRNAs, and heart disease is my next topic. The big picture: stressed immune cells release exosomes containing microRNAs that signal to the tissue cells and can induce an inflammatory response. And in particular here, I'm going to be talking about MIR-155, which is clearly getting produced in response to this virus, and especially the spike protein. The S1 subunit of the spike protein actually gets detached enzymatically and becomes free. And then it can bind to the H2 receptor because it contains a receptor binding site. And the H2 receptors are present at high levels in the heart. So what happens is that the S1 binds to and suppresses the H2 receptor. This causes angiotensin II to go up. This induces inflammation and cardiovascular disease. And then it's been shown that S1's been found both in the patients long after the virus has been cleared, and it's believed to play a critical role in long COVID, and it's also been found in the vasculature following vaccination. And then this overexpression of this particular microRNA is directly linked to worse outcomes in heart attack. And as you will see in a moment, we are finding heart disease happening in vaccinated people. Even young teenagers are dying from a heart myocarditis in response to these vaccines. So this is a paper about exosomes. And it talks about their ability to transfer these microRNAs. Of course, these microRNAs, like the messenger RNA, are fragile. They can just be broken down if they're just left open. The mRNA is very easily broken down normally, but because of the way the vaccine has arranged it, it's got it safely packed inside those particles. These exosomes can do the same thing for the microRNAs. These microRNAs are short sequences of RNA, and they have an ability to suppress selected proteins. There's lots and lots of them, like thousands of different ones. They're very complicated. And then they survive well inside the exosomes, and they facilitate cell-cell communication in a big way. And so the immune cells actually release exosomes that contain these microRNAs as signaling molecules, and then the tissue cells pick them up, and that influences their metabolic policy because of the particular microRNAs that are in the exosomes. So this is a nice paper that shows how important these exosomes are for the development of the immune response. That is what the vaccine wants to happen. You actually depend upon exosomes being transferred from the T cells to the B cells as they're trying to perfect their antibody shape. And those exosomes provide these microRNAs, and in particular, MIR-155. This one is an important one in this process, which induces the B cells to perfect their antibody response and to proliferate. So you get lots of B cells. Over here, you have a defective colony of cells that don't have this RAB27 protein that is essential for exosome secretion. So they can't secrete exosomes, and when they can't, they don't produce enough of these B cells. They're deficient. So these exosomes are really important. They carry the microRNA 155, which is also important, and they succeed in producing all these antibodies. Now, this is a paper specifically about this mRNA 155 in SARS-CoV-2, and they found that it was expressed strongly in both the original SARS and the SARS-CoV-2. But the CoV-2 had two times as high stimulation. It produced more of this interferon gamma, which upregulates the MIR155. So it was it actually had a different design in the protein sequence that caused it to be more stimulatory, to produce more of the MIR155. Here's a persistence of SARS-CoV-2 S1 proteins. This gets into the issue of S1. Spike can be broken into S1 and S2, and S1 gets released. And this is causing a lot of troubles. Many papers are showing up that are suggesting that that's a major part of the toxicity of the virus. And also, as I said, of the consequences of long COVID, when the virus is long cleared, there's still that S1 sitting around causing trouble. So it gets cleaved by the furin proteases, gets released, it gets circulated, gets picked up, and it binds to the H2 receptors and could be the cause of long COVID. So here's a nice picture of these spike proteins. Here's a cell that's making these spike proteins and sticking them on its membrane. These things get picked off. This is the S1 getting picked off. It goes and binds to the H2 receptor, and then that suppresses the H2 receptor. And you get a situation of angiotensin II getting upregulated, inflammation, thrombosis, and pulmonary damage. All of these problems can come from the consequence of S1 getting picked off from the spike protein, both in the case of the vaccine and in the case of the virus. So here was a study, 11 out of 13 healthcare workers who had detectable levels of spike protein or the S1 component in their plasma as early as 1 day and up to 28 days following their first messenger RNA vaccine. The peak level was after 5 days. So here's some data on myocarditis and pericarditis cases in VARS through June 25. And these are different age ranges, the young and the middle ages up to age 39. So none of these people are old. The number of reports on the flu vaccines over 20 years. Here, the number of cases where they had this, very, very low. Here, just in 6 months, you have much, much larger numbers of cases being reported in the US reporting system for myocarditis or pericarditis, heart inflammation, as a consequence of these mRNA vaccines. And so how is that happening? It has to do with the H2 receptors. I think it seems pretty clear to me how it could happen. Again, it has a spike S1 unit. It causes stress. It binds to H2 receptors. Angiotensin II goes up. So here's the macrophage getting stressed by the virus. Exosomes get secreted. MIR155 gets expressed. And interestingly enough, the fibroblasts take up the exosomes that were released by the macrophages, and that it causes them to suppress their proliferation and interferes with the healing process. And that can lead to cardiac rupture, and then you can die from that. And so here's a paper from 2011, long before COVID, MIR155, singled out as a microRNA that is linked to worse outcomes in heart attack. This is controls having no heart attack. This is people who died within the first 24 hours. And this is after they had some time, 2 to 7 days, they developed a large increase in microRNA 155 before they finally succumbed to the heart attack. So this microRNA is clearly implicated as a component of causing ventricular rupture and death after a heart attack or after heart disease. So exosomes, microRNAs, and prion disease is my next topic. Exosomes and Parkinson's disease. Parkinson's disease often begins in the gut as an immune reaction to prion-like proteins produced by pathogens. The spike protein is a prion-like protein. It has this glycine zipper motif. It has 5 of them. It's characteristic of prions. Stress immune cells in the digestive tract and the spleen upregulate alpha-synuclein and release it packaged up in exosomes along with foreign misfolded proteins. They travel along the vagus nerve to the brainstem nuclei. They damage the substantia nigra. They cause Parkinson's disease. And the whole process can take years or decades before symptoms appear. So here's a paper showing, arguing that COVID-19 is a perfect storm for Parkinson's. Loss of smell is an early symptom, also a symptom of Parkinson's. The virus can gain access to the brain along nerve fibers, the olfactory nerve, the vagus nerve. Neuroinvasion upregulates alpha-synuclein, and high levels of alpha-synuclein lead to misfolding and toxicity. And the dopaminergic neurons in the substantia nigra express high levels of the H2 receptor. And 3 independent case reports have described the development of Parkinson's disease immediately following the vaccine, following the disease, having had no evidence of Parkinson's before that. SARS-CoV-2 spike activates human microglia in the brain via exosomes loaded up with microRNAs. So this is an important paper because it showed these 2 RNAs, 148A and 590, as being clearly singled out as microRNAs that were expressed in response to exposure to the spike protein. And they can get internalized by the microglia in the brain, which are the brain's immune cells. And this can cause inflammation in the brain, which can lead to a neurological CNS, central nervous system damage. And here's a picture of this. The spike transfected cell produces the spike protein, produces these microRNAs, ships them through exosomes along the vagus nerve to the brain, and then it can pick it up with the microglia and end up with neuroinflammation. SARS-CoV-2 spike protein interacts with amyloidogenic proteins, potential clues to neurodegeneration. This is an interesting paper because it turns out that spike, that RBD domain, binds to heparin and to heparin binding proteins. And this accelerates the aggregation of amyloid proteins. So when you start to get spike protein becoming misfolded, it aggregates with these other proteins that are known to misfold in association with neurodegenerative diseases. And that can lead to neurodegeneration in the brain. And then here's a paper describing the whole process with a prion foreign prion protein, such as the spike, acts like a crystal, induces the misfolding of these other human proteins that are classically associated with various neurodegenerative diseases. They change from alpha helix to beta sheet configuration, and that causes these oligomers and these fibrils that are associated with many different severe neurodegenerative diseases. And so here's just a report. This is a paper that shows that there's lots of signals for Parkinson's symptoms in the response to the vaccines. And there's a couple of quotes from this paper. All the COVID-19 vaccines on the market contain spike protein or its nucleic acid sequence, creating a possible catastrophic epidemic of prion disease in the future. This analysis should serve as an urgent warning to those mindlessly following advice of politicians and public health officials regarding COVID immunization. So some other consequences. The microRNA MIR148A functions as a critical regulator of B cell tolerance and autoimmunity, a very important paper that shows that immunotolerance ensures that the immune cells can react to foreign antigens but do not attack self-tissues. Overexpression of this microRNA, which is associated with the spike protein, disrupts B cell tolerance. And that's going to cause autoreactive B cells that can cause lupus, rheumatoid arthritis, diabetes, and multiple sclerosis. Patients with lupus show increased expression of this microRNA, and it suppresses the expression of the autoimmune suppression, this guy, and of the tumor suppressor P10 and the apoptotic protein BIM, and all of these are going to cause systematically increased prevalence of autoimmune disease and cancer. And here's a paper on thrombocytopenia and microRNA 148. This is low platelet count, so the platelets are getting destroyed. 1/3 of the body's platelets are housed in the spleen. They have this receptor for immunoglobulin G, and they in these antigen-antibody complexes, such as what the spike protein would form. Activating that receptor leads to platelet activation, aggregation, and thrombosis. This particular protein here, TULA2, suppresses platelet activation, and it gets suppressed by this microRNA, leading to the aggregation that's associated with this heparin-induced thrombocytopenia in the absence of heparin. And higher expression of MIR155 in the peripheral blood monocytes of patients with this thrombocytopenia also correlated positively with a reduction in platelet count. Glioblastoma and other cancers, interestingly enough, both microRNA 148A and microRNA 590 are linked to increased bad outcomes in glioblastoma, which is the most aggressive form of brain cancer. Here's a paper about new onset neurologic symptoms and related neuro-oncologic lesions after COVID-19 vaccine. So 2 neurosurgical cases of people who had cancer, metastatic malignant melanoma and glioblastoma, and who had a very bad reaction to the vaccine that could have been related to these microRNAs that could have been delivered to the brain in spike protein containing exosomes. And here's another paper talking about multiple sclerosis associated with this one, MIR155. It's among the mRNAs that are most strongly implicated in autoimmune diseases. It polarizes the cells towards a pro-inflammatory form, strongly overexpressed in B-cell activated lymphomas, and it causes pro-inflammatory polarization of microglia in the brain to an M1-like phenotype, which is inflammatory and resulting neurotoxicity. It was upregulated 12-fold in the white matter lesions associated with multiple sclerosis. And then other diseases are linked to MIR155 overexpression, including this huge list of very severe diseases that were shown in this paper, which focused on that one microRNA. Spike antibodies and autoimmune disease. There's a substantial difference between the antibody responses elicited by the natural infection and the mRNA vaccination. And this, I think, is a serious problem. They are happy that they're seeing up to 10 times higher level of antibody expression after the vaccine compared to the disease. But we know the disease is offering very good protection. And I don't think this excessive antibody response is a good idea. I think that's where you're going to get the autoimmune diseases. The high antibody titers opens you up for autoimmune disease, especially when MIR148A is overexpressed. And this is a paper that shows huge number of proteins that bind to the antibodies to the spike protein, suggesting a potential for autoimmune disease. And they're associated with all of these diseases over here, which is a huge list of problematic autoimmune diseases that could be caused by antibodies produced against the spike protein. And this is a paper that shows a high frequency of cerebral spinal fluid autoantibodies in COVID-19 patients with neurological symptoms. And they're arguing with the potential molecular mimicry associated with SARS-CoV-2, awaiting identification, but nonetheless showing that there are these autoantibodies to the nucleus of these in the brain, targeting the brain that would be expecting, toxicity. Autoimmune hepatitis following COVID-19 vaccine, 2 case studies, 2 women, 135, 171, both of them had, there were anti-nuclear antibodies in one case. Both of them had, the second one had IgG, markedly upregulated massive eosinophil infiltration, looking like drug-induced liver injury, which has also been linked to COVID-19. Annexin A2 is a cytokine. Cytokines upregulate annexin A2 following COVID-19 and also following vaccination. It's a membrane scaffolding protein that facilitates reactions involving thyroidoxin, which is going to protect from hydrogen peroxide damage by inactivating hydrogen peroxide molecules. Antibodies to the spike bind to annexin A2, suppressing its action and therefore inducing stress, oxidative stress. And so this paper just shows that annexin A2 autoantibodies show up in COVID-19, and they are an indicator of bad outcomes. The people who have the higher levels of those antibodies are more likely to die. Reverse transcription of spike RNA into DNA. This is an interesting web page where they talk about enthusiastically about the opportunity to use these people have a specialized deuterium biomolecule stabilization system where they can put deuterium into various molecules to keep them stable. They're very excited about potentially using this for the RNA vaccines. The impact of COVID-19 has heightened the urgency of our technology, and our scientists are starting to create results. And this was in November 17th of 2020. We don't know if this is actually being used in the current vaccines. They wouldn't necessarily have to list it, but it may well show up in the future. And I think it would be bad to and it makes the RNA more stable, which makes it more likely to stick around and more likely to get converted into DNA, which is possible through this, for example, through the sperm. And this was a paper that we talked about, Greg and I talked about in our paper, how sperm can insert DNA from RNA into the fertilized embryo and transfer it to the offspring. It's quite remarkable. They have a reverse transcriptase activity that can take exogenous RNA molecules, such as the messenger RNA in the spike protein in the vaccines, and turn them into cDNA copies. And then they can transfer from the sperm to the early embryos at fertilization. And then they're passed on from the embryos. The fetus has these same, in these plasmids, they have these plasmids containing the cDNA that can be, for example, coding for the spike protein. And they can pass it on to their offspring. And it could even possibly get incorporated into the nucleus to become a permanent part of human DNA. And sperm-mediated reverse gene transfer happens when these cells are incubated with exogenous RNA molecules, which is exactly what these vaccines are. Polymerase theta is an interesting DNA polymerase, which has been found only recently to be able to have a very good skill at converting RNA to DNA, just like the reverse transcriptase that the HIV virus produces. And so it looks unique among the human polymerases in this capability of converting RNA to DNA, and it shows up in cancer cells and protects them it interferes with cancer cell treatment. Summary part 2, the novel vaccine technology for COVID-19 prevention is untested and will potentially cause devastating neurodegenerative, autoimmune, oncological, and vascular diseases in the vaccinated population. Primary mechanism may be through the release of massive numbers of exosomes containing spike protein and specific microRNAs. Antibodies to spike protein also bind to many human proteins associated with diverse autoimmune diseases through molecular mimicry. There's real potential for the messenger RNA to be converted to DNA and sustained in plasmids in germ cells or cancer cells long term. Continued production of spike protein would likely enhance symptoms of autoimmune and protein misfolding diseases. There's even the possibility of transfer to future generations and integration into the human genome. Thank you very much.